Hello everyone. In this video, we are going to discuss about the chapter Molecular Basis of Inheritance, and part 10 here focuses on transcription. This is again one of the very important processes that takes place inside the body of an organism. You remember the central dogma that we learned was DNA is converted to RNA, and then RNA gets converted into protein, through highly sophisticated processes, all these processes. And in the last part of this chapter, we learned about replication. That is when more DNA is formed, that is one of the major fundamental processes. And now transcription, which is formation of RNA from DNA. So this process here is transcription. And we'll see how it is also important from the point of view of NIT examination as we progress through the video. So the process of copying genetic information from one strand of DNA into RNA is termed as transcription. Okay, so only one of the strands, like DNA we all know is a double helical molecule, but only one of the strand and only a certain set of this strand is converted to RNA. And this process is called as transcription. It is generally considered to be the first step in gene expression. You remember, right, that DNA has genes, and how are these genes expressed? These genes are expressed in the forms of chains of amino acids which form the protein. So this is how a gene is expressed, in the form of protein. So the first step in gene expression is conversion of genes on the DNA to RNA. Okay. The principle of complementarity governs the process of transcription, except adenosine now forms base pair with uracil instead of thymine. By now we all know what is the principle of complementarity, right? That if we know the sequence of one strand of DNA, we can easily determine the sequence of other strand, and we have seen this multiple times in the previous parts. In case of transcription also, this principle of complementarity is followed. In case of DNA, what happens is, we know that adenine pairs with thymine and cytosine pairs with guanine. In case of RNA, however, when one strand of DNA is converted to RNA, there is again the same principle of complementarity. If you know the sequence of DNA, you can easily determine the sequence of RNA. The only difference here is that adenine now pairs with uracil instead of thymine, in DNA. Cytosine again pairs with guanine. So this remains the same. The only difference is here. Okay, but the principle of complementarity governs the process of transcription as well, just like replication. In transcription, only a segment of DNA and only one of the strand is copied into RNA. We just discussed that, only one of the strand and only a segment of DNA is copied to RNA. The mechanism of transcription is basically similar to that of replication, but it differs from it with respect to the following. There are always some differences, What are the differences? The precursors are ribonucleoside triphosphate rather than deoxyribonucleoside triphosphate. And what is the difference between ribonucleoside triphosphate and deoxyribonucleotides? It is the difference of the pentose sugar. You remember this, right? Ribonucleoside triphosphates that form the RNA have the sugar ribose. It has an extra 2' OH. It also makes it like less stable and more degradable. So this pentose sugar in case of RNA has ribose, and therefore the precursors are ribonucleoside triphosphate. That consists of ribose, it consists of phosphate group, and it consists of nitrogenous bases. So this is the composition of ribonucleotide triphosphate rather than deoxyribonucleotide triphosphate. The only difference is that the sugar that is there in DNA is deoxyribose. Okay, that is the only difference. In replication, once the process has initiated, the entire DNA of the organism is duplicated. However, in transcription, only a particular DNA segment is transcribed. Not all of the DNA strand is converted to RNA, only a segment of the DNA is transcribed. Only one strand of the DNA is used as a template for synthesis of complementary in any given region. So we know that not both, but only one strand of DNA is used for transcription. RNA chains can be initiated without any requirement of a pre-existing primer strand. If you went through the last part of this chapter, like replication, we saw that replication of DNA requires an RNA primer. Okay, so an RNA primer must be present that is always synthesized in 5' 3' direction, and only then is DNA polymerase capable of adding nucleotides to form the DNA molecule. You remember this, right? If you don't remember, just go and watch the video again. So this was in case of replication. However, in case of transcription, there is no requirement of a pre-existing primer strand. Okay. And transcription is carried out by DNA dependent RNA polymerase. Like DNA, replication is by DNA polymerase, transcription is by RNA polymerase. Transcription unit. What is a transcription unit? The segment of DNA on which one primary transcript or the initial RNA molecule is transcribed is known as a transcription unit. So it is basically, a segment on DNA. So this is one strand of the DNA that will act as a template for the mRNA. So this segment of DNA on which one primary transcript or the initial RNA molecule synthesized from DNA is transcribed. So this segment here is called as a transcription unit. The primary transcript in prokaryotes is generally equivalent to the mRNA, messenger RNA. You remember this, right? RNA are 3 types and the one that helps is in messenger RNA because it acts as a messenger between DNA and proteins. So the primary transcript that is formed in case of prokaryotes is equivalent to mRNA molecule, while in eukaryotes, things are always a bit more complex. In eukaryotes, the primary transcript must be processed to form mRNA and hence it is often called as pre-mRNA. So, the RNA that is formed from here in case of eukaryotes is called as pre-mRNA and then it must be processed. And we'll see what kind of processes are going to take place. It must be processed to form the proper mRNA transcript. However, in case of prokaryotes, this is equivalent to the mRNA. The first transcript that is formed is equivalent to mRNA. And what are the components of a transcription unit? It contains 3 regions, that is promoter, structural gene and a terminator. So here we see a diagrammatic representation of the transcription unit and the 3 components are promoter, the structural gene and the terminator. Okay, this is very important. So what is promoter? As the name suggests, promoter is something that will promote the transcription. So promoter will be a specific sequence on the DNA that will determine the transcription start site, that this is where the RNA polymerase has to bind and start the process of transcription. Then there is second thing that is called as the structural gene. This structural gene basically contains the sequence of nucleotides that will determine the sequence of the pre-mRNA or the mRNA. Okay, and this is here, one strand is template strand, other strand is coding strand. So now it is very important to know what is a template strand and what is a coding strand. So template strand is the strand that acts as a template to the mRNA. Okay, so the mRNA that will be formed will have a sequence that is complementary. That is complementary to the template strand. Okay, it should be very clear. The mRNA that is formed will have sequence that is complementary to the template strand because template strand is the strand that will provide the sequence for transcription. And the other strand is the coding strand. It is called as coding strand because this has sequence similar to mRNA. So this has the sequence that is similar to mRNA. The only difference here that is very important is what we all know, that adenine will pair with uracil. Okay, so instead of thymine on DNA, there will be uracil on the RNA. So this is the only difference. Otherwise, the whole sequence will be the same as the coding strand. So mRNA sequence is exactly like the coding strand. The only difference is that instead of thymine on DNA, we have uracil on RNA. And the template strand acts as the template and has the structure complementary to the mRNA. And the third sequence is terminator. As the name suggests, it causes termination of transcription. So gene on a transcriptional unit. A gene is a functional unit of inheritance. We all know this. What is a cistron? A cistron is defined as segment of DNA coding for a polypeptide. The structural gene in a transcription unit could be said as monocistronic or polycistronic. So what is cistron? It is defined as a segment of DNA. So for example, this is one of the strands of DNA. Let's say for hypothetical and explanatory reasons, this is one strand of DNA as long as it is. So cistron is a segment of DNA that codes for a polypeptide. Okay. So this is very important, that cistron always and only has sequence that codes for a polypeptide and nothing else. The structural gene in a transcription unit could be monocistronic. What is monocistronic? Mono generally means one, So the sequence that codes for only one polypeptide, which is generally the case in eukaryotes, because one gene or one cistron codes for only one polypeptide. However, it can also be polycistronic, which is generally the case in prokaryotes. Okay. So in prokaryotes, one cistron can code for multiple polypeptides, but in eukaryotes, one cistron will code for only one polypeptide. This is important, monocistronic and polycistronic. The eukaryotic genes are split as their coding sequences called as exons for expression sequences are interrupted by intervening sequences called as introns for intervening sequences. We'll go one by one. The eukaryotic genes, they are split, For example, this is the DNA. So in eukaryotes, it is not that this is the DNA and this is exact, like let's say this is a cistron on the DNA and this is the RNA. That is not the case in case of eukaryotes. In case of eukaryotes, what happens is, for example, this is the DNA. This is the sequence that codes for a polypeptide, or it is a cistron. Okay. Something like this, for explanatory reasons, will be the RNA. You see, there are two things. One is this bold red that I am drawing and one is small red. So this bold red thing that I am drawing is called as exons. Exons for express sequences, and the less bold lines are introns. So introns are basically intervening sequences in between the exons, and these introns must be removed for the exons to come together and form the proper mRNA. Earlier we talked that in case of eukaryotes we have a pre-mRNA. So this is the pre-mRNA, and one of the important things that must take place is slicing of these introns here to form the mRNA. We'll see it soon in more detail, okay? In a mature or processed RNA, introns are not present. So they must be removed for a mature RNA or mature messenger RNA to be formed. So this is a gene structure in case of prokaryotes. This is the DNA. And this strand, for example, here has the coding region. So this is regulation of transcription. We have the promoter. Okay. And then is the regulation of translation. We'll see about this in the later parts, So we can for now ignore this. And then there is this coding strand, and then there is signal for termination of transcription. So in case of prokaryotes, what happens is there is this DNA that forms the primary transcript. So this is here the mRNA. And in case of prokaryotes, this primary transcript is equivalent to the mRNA that is then converted by translation to form the polypeptide chain. So the only thing we have to remember here is when transcription takes place in case of prokaryotes, the primary transcript that is formed after transcription is equivalent to the messenger RNA. There is no post-processing that is needed, and ultimately it gets translated to form a polypeptide chain. But in case of eukaryotes, it is a little bit more complex. How? We will see it. Again, here there is this regulation of transcription, and then there are these exons. Exon 1, exon 2, exon 3. And these exons are the only sequences that code for the mRNA, and they are bound by intervening sequences called as introns. So when DNA is converted to a primary transcript by transcription, there is exon, then there is this red intron, then exon, then red intron, then exon, and then further sequences. There is a cap and there is also a poly A tail. So this is what is the composition of a primary transcript, and this cannot at this stage code for a polypeptide. It must go post-processing to form mRNA. And what are these post-processings? These post-processing is removal of these introns, So these introns form like a splice structure, and then they form a spliceosome complex. Okay, so it is important to remove these introns. Removal of introns by formation of spliceosomes. So this spliceosome complex helps in removal of these introns from here. And once these introns are removed, it forms the mRNA that can be now translated into a polypeptide chain. Okay, so the difference is very important here. In prokaryotes, the primary transcript that is formed is equivalent to mRNA. However, in eukaryotes, it must undergo post-processing. And post-processing is removal of introns by formation of a spliceosome structure. So then we see the process of transcription. Although the basic features of transcription are same in both prokaryotes and eukaryotes, but some features such as the promoter sequence, they are different. And we'll first see transcription in prokaryotes, the lower organisms, that is generally much simpler. The DNA-dependent RNA polymerase of prokaryotes catalyzes synthesis of all types of RNA. There are 3 types of RNA, 1, 2 and 3. So the DNA-dependent RNA polymerase for prokaryotes can cause synthesis of all these types of RNA, and the process of transcription can be divided into 3 stages. It is initiation of the new RNA chain, elongation of chain, and termination of transcription and release of the nascent RNA molecule. Okay, so the steps are like the same. There is initiation, there is elongation, and then there is termination. So initiation. RNA polymerase recognizes a specific site on DNA which is upstream from the gene that will be transcribed, that is called as the promoter site. Now, if you remember, the structure of a transcription unit on the DNA molecule, we always said that there was a transcription start site and there was a promoter region. So, RNA polymerase recognizes a specific site on DNA upstream. So, what is upstream? For example, this is the cistron here, or this is the gene that has to be transcribed. Upstream will be any sequence that is on the left. Downstream would be any sequence that is on the right. Okay, so these are something important things you need to know. So upstream from the gene that will be translated called as the promoter site, this site here, and then unwinds DNA locally by recognizing the -35 sequence. So this gene sequence will be 0. 35 nucleotides upstream of this sequence, which is the -35 sequence, will be recognized by the RNA polymerase, and unwinding of the DNA will take place, because DNA is double helical structure. So it is this recognition of the -35 sequence by the RNA polymerase, which is called as the promoter site, that will unwind the DNA. RNA transcription requires enzyme RNA polymerase, DNA template, all 4 types of ribonucleoside triphosphates, which are ATP, GTP, CTP, UTP, divalent metal ions, magnesium or manganese as cofactor, and the Rho factor. So you must know that all these components are required for the process of RNA transcription. most of which we already know. We know that it requires RNA polymerase, of course it requires DNA templates, or 4 types of ribonucleoside triphosphate. Then there is divalent metal ions, magnesium ions and manganese ions, and then there is this Rho factor. RNA polymerase can initiate transcription at specific DNA sequences called as promoters, and we saw, there is this promoter site that has the -35 sequence. So RNA polymerase can initiate the transcription by recognizing this -35 sequence. It then produces an RNA chain which is complementary to the DNA strand used as the template. This also we know already. So first we talk about the initiation. So RNA polymerase, and here we are talking about prokaryotes, So RNA polymerase has 5 polypeptides, and these are the 5 polypeptides. The sigma, B, beta dash, alpha and omega. Okay, so these are all the 5 components that the RNA polymerase have. Now, the chain of beta, beta dash, alpha and omega constitute the core enzyme. Okay, so the sigma factor is not a component of the core enzyme, and rest all constitute the core enzyme. Sigma factor recognizes the promoter region while the remaining enzymes take part in transcription. Okay, so you see here this is the RNA polymerase. This is the entire RNA polymerase, but you see the sigma factor is alone. Why is this sigma factor alone? There are 2 reasons, The first reason is that sigma recognizes the promoter region, while the remaining core enzymes, they take part in transcription. So sigma factor kind of acts alone. Okay, and then the beta, beta dash, alpha and omega, they constitute the core enzyme. So you see this dark orange structure here is the core enzyme, and there is this sigma factor that are responsible for initiation. The sigma factor is capable of recognizing, the promoter sequence. The next is elongation. So once the sigma factor has recognized the promoter sequence and caused unwinding of the DNA, here you can see, unwinding of the DNA, then the sigma factor releases and the only core enzyme is now present, Okay, so this is important that RNA polymerase has a core enzyme and a sigma factor. The sigma factor will recognize the promoter sequence. It will cause unwinding of the DNA and then it will release, it is no longer required. And then the core enzyme participates in the process of elongation. Core enzyme contain polymerization catalytic unit within the beta subunit. So the beta subunit is the catalytic polymerization unit. The first nucleotide in RNA transcript is usually either G or A. So this is the triphosphate, that is present initially. So this is important. They can sometimes ask a very easy and direct question in the NIT examination. The first nucleotide in RNA transcript. So it is usually G or A. RNA polymerase then synthesizes RNA in 5', 3' direction using ATP, CTP, GTP, UTP as precursor. Okay, so these are all the ribonucleoside triphosphates that are used as precursors, and the elongation will take place by RNA polymerase. And then there is this process of termination, because only a segment of DNA is being transcribed. So definitely the process of termination has to take place. So transcription continues till a termination sequence in the form of GC rich or palindromic region is reached. This is very important, So this GC region or a palindromic region is very important. So this acts as a termination sequence. Okay. DNA of this palindrome is self-complementary and base pairs internally to form a hairpin structure. So, it will be something like a GC rich sequence and it will look something like this, a hairpin like structure, and here will be like multiple GC, GC, and this will be like some other small sequence, for example, A, T, C or something like that, G, but here will be the GC sequence that will base pair with each other, and this is the palindromic sequence. Okay, it will form a hairpin structure. You see here it will form a hairpin structure. Termination sequence is located downstream towards 3' end of the coding strand. You remember I told you previously in this slide, for example, this is the DNA sequence and this is the gene sequence that or the cistron that codes for a polypeptide. Any sequence that is below this sequence is called as upstream sequence, okay? And anything that is on the right is called as the downstream. So the termination sequence is located downstream, of course, because you want the entire gene to be transcribed. So the termination sequence will definitely be downstream towards the 3' end of the coding strand. And rho factor helps in termination of RNA synthesis. So sigma factor helps in initiation and rho factor here helps in termination. So this rho factor will come and bind to the core molecule and will cause its release, and ultimately it will release the RNA transcript. Okay, so there are 3 steps: initiation, elongation, termination. The sigma factor and the core RNA polymerase start the initiation by recognizing the promoter sequence, which is -35 sequence. And then the sigma factor will release out, the core unit will cause elongation by RNA polymerase and adding ribonucleoside triphosphates. Once the palindromic sequence or the hairpin loop GCH sequence is reached, the rho factor will now cause termination and release of the RNA polymerase. So this is how transcription takes place. So that was quite simple. It was in eukaryotes. It was in prokaryotes previously what we saw. In case of eukaryotes, the main principle is quite same. It is just that we have a lot of factors involved. Okay, so for initiation, the RNA polymerase cannot initiate transcription by themselves. They require assistance by transcription factors. An initiation complex is formed by transcription factors before the RNA polymerase binds and initiates transcription. Okay, so for example, this is the RNA polymerase that we have. There are multiples of transcription factors that will bind in a specific way. They will all bind together. They will form another kind of structure. And then this entire structure will then cause the process of transcription in case of eukaryotes. It is much more complex, that you require much more transcription units. The 3 polymerases require different promoters and transcription factors. Okay, so in case of eukaryotes, it is much more complex. Synthesis of nuclear pre-mRNA involves RNA polymerase 2 and it is found to transcribe most of the eukaryotic genes. So you have to remember this, that RNA polymerase 2 causes the synthesis of nuclear pre-mRNA. And then next is elongation. RNA chain elongation takes place in eukaryotes by the same process as in prokaryotes. Once the eukaryotic RNA polymerase have been released from their initiation complexes. Okay, so in case of prokaryotes, we saw that once the RNA polymerase bind, it causes elongation by adding what? By adding ribonucleotide triphosphates. Okay, and this ribonucleotide triphosphate has the ribose sugar. So that is how similarly elongation takes place in eukaryotes. When the growing RNA chains are only about 30 nucleotides long, 5' end of eukaryotic pre-mRNA are modified by addition of 7-methyl guanosine triphosphate caps, and the process is called as capping. So there is like a kind of processing of this mRNA chain that takes place. And these processings are important for the later steps, for translation. So let's just look at it again. When the growing RNA chain are 30 nucleotides long, when elongation is taking place and only 30 RTPs are added, therefore the RNA chain is 30 nucleotide long, the 5' end of the eukaryotic pre-mRNA are modified by addition of 7-methyl guanosine triphosphate caps. It is again a very important question, for NIT examination, because they can really ask what does the cap contain. It is 7-methyl guanosine and this process is called as capping. It has an unusual 5' triphosphate linkage and 2 or more methyl groups. Okay. So generally what is the linkage? That there is always the linkage of 5' 3' linkage, okay, and then nucleotides are added on this 3' OH and 5' phosphate. So the linkage that is always present is 3' OH and 5' phosphate. However, in this case, there is a 5' 5' triphosphate linkage and 2 or more methyl groups are present. And then these caps have two main functions. They are recognized by protein factors involved in initiation of translation, okay, formation of protein. That is why this cap is added here. And they protect the growing RNA chain from degradation by nucleases. So these are also again, very important questions from the point of view of examination as well. So what are the main functions of the cap? The main functions are that they are recognized by protein factors involved in initiation of translation. For translation, for formation of protein, there are certain factors and we will have a look about that in the later parts of this chapter as well. And the second most important is that the growing RNA chain, it protects it from degradation by nucleases, because RNA we all know, it is not very stable. It can be easily degraded. So this 5' methyl guanosine cap protects the growing RNA chain from degradation by nucleases. So here is the process of transcription in case of eukaryotes. So this is the RNA polymerase, with a large number of transcription factors that recognize the site and then it causes elongation. And when the growing chain is only 30 nucleotides long approximately, there is this 5' methyl guanosine cap that is attached. Okay, so this is the 5' cap attached, and what are the functions of this cap? They help in initiation of translation and they provide protection to the RNA. Okay, protection against degradation. So this is very important, okay. So then this cap is formed that has like this 5' cap. This mRNA chain that is formed has a 5' cap, but it has introns. Okay, so exons were the express sequence and introns are the intervening sequence, and these introns must be removed. So these introns are removed, by forming a spliceosome complex, and then a poly A tail is attached, and this process of attaching of multiples of poly A tails is called as polyadenylation. And this is again important, because the poly A tail also serves similar function as the cap. For example, it also protects against degradation. So it protects the mRNA. RNA is very unstable. We all know that by now. So this polyadenylation, which is just addition of multiple adenines towards the end of the transcript, and the cap, they provide protection against degradation. And then this is the final mRNA product that is formed. So you see, in case of eukaryotes, there are multiple extra things that are happening. What are those? The first one is that here it is the RNA polymerase that has multiples of transcription unit and they must be assembled first. So first what happens is, this is the RNA polymerase and it assembles with these multiple transcription units, and only then it goes and binds to the DNA. When the growing chain is approximately 30 nucleotides long, then there is addition of this 5' cap that also acts as a site for translation initiation, and it provides protection against degradation. Then this, primary transcript has introns that must be removed. They are removed by the process of RNA splicing, and then poly A tail is added, which is multiples of adenines that are added. They can be up to 200 or even more adenines that can be added, and they also protect the mRNA. And then ultimately the final mRNA is formed in case of eukaryotes. So the process of transcription in eukaryotes, the principle is the same, but there are certainly much more differences. So then is termination, So tailing. Adenylate residues, we discussed about the poly A tail. So adenylate residues, approximately 200 to 300, are added at the 3' end of a template in independent manner. What does it mean in an independent manner? It means that the sequence is not present on DNA. The DNA only codes for the polypeptide sequence. It does not DNA does not code for poly A tail. Okay, this is important. DNA does not encode for the poly A tail. Therefore, the poly A tail or the adenylate residues are added at the 3' end in an independent manner. The process of addition of poly A tails to the eukaryotic mRNA is called as polyadenylation. Fully processed RNA, now called as mRNA, which is transported out of the nucleus for translation. So now these mRNA are transported out of the nucleus for translation. So the process of transcription takes place where? In the nucleus. Why is that? Because DNA is present in the nucleus. And once the mRNA is formed, it is then transported outside the nucleus and where the translation takes place. Okay, so this is also important. There is this thing called as compartmentalization. The process of transcription takes place in the nucleus and translation takes place outside the nucleus. Then RNA splicing. We also studied about RNA splicing, no? So intron sequence are removed by the process called as splicing and ligates the end of exon sequence together. For example, we earlier saw similarly, like these are the exons, the small ones are the introns, exons, introns, and then there is splicing. So these 3 exons are joined together, and this is what the final mRNA transcript will have. So this chapter really focused on another important topic, that was transcription, and you must know, all the processes that are taking place. So they can really ask you questions, especially, around the polyadenylation, around the 5' gap. How are there differences between eukaryotic and prokaryotic transcription? And what does a transcription unit contain? It contains a promoter, it contains a gene sequence, and then it contains a terminator. Also, you must try to know the difference between the coding strand and the template strand. Okay, this is important. The template strand basically has the sequence that will be complementary to mRNA, whereas the coding strand will have the sequence similar to the mRNA. The only difference is that in case of RNA, adenine pairs with uracil instead of thymine. Okay. Thank you for watching the video.